first speaker this afternoon is an old-timer in the ARDD. He was with us even when we were in Basel. But it's the second time you're with us, so let's put it that way. But we're very happy that you're here, Pekka. So please take the stage and let us know something about stem cells.
Thank you so much, Morten, and thank you also for singling out Tim as a largest individual. I don't see Tim anymore. Maybe he left already. Thank you to all the organizers for inviting me, and Denmark always has the most amazing venues in meetings. So of course happy to be here. Our lab indeed is very interested in stem cells and their role in aging, and this crowd does not really need an introduction to aging. All I want to do here with this slide is to point out into this reduction or decline in function of various tissues that is ongoing at the same time as many of those diseases that maybe most of the crowd here is concentrating on is ongoing. And whether this is actually the cause of some of those diseases or not, of course, that's another debate. But still, because of that functional decline that goes on with most of our tissues, we're interested in the cells whose job it is to maintain the tissue function throughout our life. And that's, of course, the tissue-resident stem cells, also known as adult stem cells. We find those in most of our tissues undergoing some level of renewal and remodeling of tissues. And the stem cells of the tissue do it so by conducting two tricks. They can either make more stem cells, aka self-renew, or then differentiate into the functional tissue cells of that tissue and keep us alive. And of course, this balance needs to be very carefully controlled, because too much of self-renewal drives cancer risk up, and whereas too much of differentiation basically reduces the number and function of stem cells in the tissue, which then is one of the hallmarks of aging, actually. Now, because it is such an important job to make those decisions between the two fates, it's good that the stem cells get a little bit of help for this. They are continuously controlled and regulated by their surrounding cells, and multiple other factors that I don't really have time to go into about this, but jointly, collectively, something referred to as stem cell niche is regulating the stem cell function, giving them also, for example, nutrients and support and protection, for example. As you well know, many things can go wrong during aging process, during the aging, biological aging, and these. I'm not going to be going into any details of which ones maybe would be more relevant than others. Suffice to say that jointly these changes that occur on a cellular level are considered to then alter this balance or hamper the processes that I just talked about, ultimately compromising the tissue renewal in the future, or the differentiation becoming faulty and therefore the functional decline. But the question in the field really currently still is, are these cellular level changes, damages, are they actually mainly taking effect via the stem cell directly, or are they functioning or altering the tissue function by altering the niche guidance, the niche support for the stem cells? And we think this is a very interesting question, relevant for also many of the aging-related problems. And we study this, for example, in the intestine. Now, I know that the intestine and the aging of the intestine is probably something that does not keep many of you awake at night. But let me assure you that intestine does age. Whether the actual the function that everybody thinks when you think about the intestine, the food intake and the absorption, whether that actually is challenged, that's a bit of a out there for a debate yet. But what definitely is changed is the barrier function. So the intestine old intestine is less capable of keeping the bacteria out, if you will. And of course, we have the whole microbiome living in the intestine, so it's a big problem for the older individuals. But maybe the most relevant one for the aging biology is the last one here, the reduction in regenerative capacity of the intestine. It's a very rapidly renewing tissue. All of you will have a new intestinal epithelium. That's what I'm referring to, the single monolayer of epithelial cells lining your whole intestine. That is renewed basically every 4 to 5 days, so that you have a new intestine next week. This makes it also extremely sensitive for any problems in this fast ongoing renewal that is ongoing. Meaning that dosing of various chemotherapeutic agents, for example, will have an impact on the tissue renewal of the intestine. And since the renewal and the regenerative capacity of the old intestine goes down, as shown in this graph here, that we published a little while ago, the elderly patients undergoing chemotherapy, for example, have a much tighter, narrower therapeutic window for many of the agents that we are using for targeting cancer, for example. Now, the cells responsible for this absolutely staggeringly fast renewal of the intestinal epithelium are found here on the bottom of the crypts, as we call them. The villi are the business end of absorbing nutrients. But this is where the stem cells are. These green little conical cells here are the stem cells, marked by expression of LGR5, made famous by Hans Clevers and co-workers already a few years ago. And they are intermingled here between these red cells, the lysozyme-positive Paneth cells, who are double agents. They are part of the innate immunity, secreting multiple different antimicrobials, keeping the crypt basically sterile, which is pretty amazing when you think of the microbiome being out here immediately close by. But they also supply the stem cells with a slew of important factors, absolutely crucial factors for the stem cell self-renewal and proliferation, including, for example, ligands, delta ligands for the Notch signaling in the stem cells and Wnt and EGFs. There's certainly other cell types in the niche that are important. I'll come back to those a little bit later. But on this epithelial niche, the stem cells and the Paneth cells we can easily isolate and put them next to each other in vitro culture. And what comes out of this is these organoids. I'm sure many of you have heard about them as well. Again, this is a technology that was pioneered by Hans Clevers and co-workers, Toshi Sato in his laboratory most importantly. And this is basically now new intestine growing on a plate, coming out from one stem cell, one Paneth cell finding each other and starting a new intestine. It's a closed system, but still these are the crypts, and basically the villus region is here in the center. And this gives us a very good idea of the capacity of this given cell pair to also form new intestinal epithelium. And we've taken a full advantage of this system over the years. The first one that we actually worked on was to study how does the calorie restriction, something that here to this crowd, of course, is a pro-longevity treatment and very interesting as such, but also it is the intestine, it is remodeled according to the food intake. So we were interested also from that aspect, how does calorie restriction actually change the system and remodel the intestine. And what we found was that when we isolated the Paneth cells and the stem cells, either from ad libitum fed or calorie restricted animals, and combined them in all four ways, that the stem cells basically do not sense reduction in nutrients directly. But instead, the calorie restriction changes the ability of the Paneth cells to boost and help the stem cells to generate new intestine very dramatically, as seen in here. And this is all because of Paneth cells apparently being hypersensitive for nutrient levels, and the TOR activity is quickly inhibited when the food intake is, in this case, was 70%. So 30% reduction, this results in, via mechanisms that we don't really understand, in expression of something called BST1, which is an ectoenzyme on the outside of the Paneth cells, producing cyclic ADP ribose from NAD. Again, we don't actually know how cyclic ADP ribose does it, but it is taken up, we know that, into the stem cells next door, and there it drives self-renewal. As I said, we don't know the mechanisms behind the self-renewal promoting behavior of cyclic ADP ribose, though. But in effect, what does this mean? It drives the pool of stem cells up in the intestine during calorie restriction. So you have more stem cells when you're fasting, and this comes at the expense of differentiation. So you actually put less cells to the differentiating axis. Therefore, the villi are actually getting shorter and reducing the very expensive to maintain epithelial real estate because the food is not there. When the food comes available, you quickly then differentiate the stem cells and extend the villi again. Now, that was, let's say, aging related. What about actually during aging? This is a question that was tackled by an absolutely just amazingly talented PhD student in my laboratory, the very first one of them, Nalle Pentin-Mikko. He isolated again now the Paneth cells and stem cells, this time from young or old mice. Old is 24 months in here, and young is 3 to 4 months. And with young and young combination, there was this baseline of epithelial regeneration that he could see in vitro happening. And sure enough, when the stem cells came from an old animal, this went down. But what Nalle focused on was this drop here, that when the young stem cells were next to an old Paneth cell, to a next niche cell, they also were not really performing so well. So he profiled what's going on with the old Paneth cells and found that they are expressing a high amount of something called Notum. And this Notum was interesting because it's actually an enzyme, deacylase, that can cleave off lipid moieties from Wnt ligands. And I already told you that Wnt ligands are one of those key factors maintaining self-renewal of the stem cells. So what happens during aging is actually that TOR becomes chronically activated in the Paneth cells. This we don't understand why. It is not dependent on the weight of the animals. It does happen independent of that, and Paneth cells have less of a sensitivity to regulate their TOR activity when in an old intestine. The high TOR activity in the Paneth cells then, via alleviating the inhibition of PPAR alpha on Notum, results in increased production of Notum, and Notum then basically kills the niche in the old intestine for the stem cells. So it goes and chews off the lipids from the Wnt. It doesn't matter how much Wnt there are. Now the stem cells are not seeing them. They're not active anymore for themselves. Now, Notum is an enzyme, as I said, so you can also target it. And we were very lucky to come across Ben Kravat's laboratory targeting this class of molecules while working on this story. And they had a small molecule inhibitor called ABC99 in their pipeline. We got our hands on it and conducted experiments where we took young and old animals, induced a mucositis in them, exactly what I was referring to earlier on, giving 5-FU, a common first-line chemotherapy therapeutic, to the mice, causing a drop in weight. So you can see in the young animals here, you have about 2% weight loss, but in the old animals, you have a much bigger loss of weight with the same dosing per gram of mouse, exactly highlighting this inability of the old animals to recover from damage to the intestinal epithelium. But when we then gave these mice 1 week of prophylactic Notum inhibitor, ABC99, therefore allowing let's call it rejuvenating the niche for the old stem cells, now the mice lost much less weight and were happily joining their young friends after this regimen. I won't have time to go into the details of it. Notum is an interesting factor for many reasons. One is that it's actually hijacked by cancer in early stages in the colorectal cancer case. APC loss is the most common reason for having colorectal cancer, and in early phases of adenoma formation, APC negative cells are expressing a lot of Notum, and therefore actually biasing the clonal competition in their own favor against the wild-type stem cells, and this is the way that they take root in the crypt. Otherwise, they are basically washed away, washed out, and becoming feces. And we can inhibit this with the APC99 that I already told you about. So this has some therapeutic potential going forward. Now I'm going to switch gears a little bit and go outside of the epithelium. How much time do I have, Morten? 5? Still, any idea? 3, all right. So this is not going to be even close to applications. But I want to show you a story that kind of, to us at least, exposed a new way of what's wrong with the old tissue. So we were interested in looking outside of the epithelium, and we're considering what could go wrong or what's changing in the extracellular matrix, the omnipresent matrix in all of our tissues, and we started conducting these decellularization protocols where this is now the intestine that doesn't have any cells anymore. This is the ECM, so this is where the crypts used to be. This is in form of villus. They are kind of shriveled up, but you can then go and slap on epithelium and grow it back on. Onto this ghost of an intestine, if you will. And this is work conducted by Sharif Iqbal in the laboratory, another fantastically talented PhD student. And what Sharif did, he did exactly our bread and butter experiment, where he mixed and matched, took young epithelium, put it onto old ECM, asking what's happening. So if you look here, young on young, this is the level of ability of the epithelium grow on young ECM. But if the same cells are now on old ECM, they are much less capable of creating new intestinal epithelium and covering real estate. Sorry. So he profiled what's going on and figured out that in just 7 days, if the young epithelium is exposed to old ECM, in about 7 days, it will harbor about 30% of those transcriptional alterations that you find in normal aging intestinal epithelium. So comparing young and old epitheliums, what's different there? About 30% of those changes are found induced by something that is in the ECM. And the main feature of that really was that jumping out from these lists was high TGF-beta signaling. I don't have time to go into the details of it, but because of that, we then conducted proteomics on the ECM and found a list of candidates. Well, short list 3 to be exact, and one of them was this short leucine-rich repeat protein or proteoglycan asporin. Which is previously linked to modulating TGF-beta signaling. It goes nicely up with age, so that was interesting. This is in collaboration with Alessandro Ori's laboratory in Jena, this mass spectrometry. And it also is expressed by the very right cells, if you will, to have an impact on the epithelium. It's the pericryptal fibroblasts here, these dark dots here. So we made our own asporin and sprinkled it on organoids to ask what does it do. And curiously, it caused this kind of a spheroidal growth marked with SCA1. And that caught our attention because of a couple of high-profile papers recently showing that this is part of, this is indicative of something of a fetal state, which means that epithelium, when it's damaged in the intestine, it goes back in time into something of a fetal, semi-fetal state for transient expansion and real estate cover. If there's ulceration, it's quicker to cover it in this fetal state. And then it after this transient phase, goes back into differentiating normally. This is done by Ofer Klein's lab and Kim Jensen, local here. So I need to speed up a tiny bit. What we are pretty sure is that asporin is involved with this because of transcriptional profiles and because of the effects of transient asporin inducing regeneration. I want to race so that I don't miss my point. So because of these reasons, we targeted asporin. We actually made a mouse model to knock it out from the mesenchyme, and then again did these 5FU experiments. Wild-type animals bounce back with their weight after the 5FU being treated here. Aspirin being knocked out from the mesenchyme do not do that, and we think it's because they cannot engage this fetal program, this transient going back in time in order to heal the ulcers that are forming at this moment. And if you look later on, so this is the 3-day point that I just showed in facts. It the transient induction of the fetal state in wild type here. None of that happening in the SKO mice, which is the targeted mice. But it comes up later when all of this has subsided in the wild types. That's when the targeted mice have it. So it's not like this is the only factor. Aspirin is not the only factor driving this fetal state up. But it seems to be, if it's not there, it is delaying this. And what does this mean for the aged animals now? I said that there's more of aspirin in the old intestine. We just knocked it out, and it was problem. What's going on with the old then? Here's the curve for the young, and here's the old animals. So they don't recover from the same dosing here, and we think this aspirin-mediated program is involved, because if you look here in the young, you have this transient, but it's more or less about 10% of cells turning into fetal state, and then they go away by day 5, whereas in the old, you drive it up at day 3, insanely high compared to the young, but more importantly, it just keeps on going. So we never resolve this fetal state. Therefore, what we actually think we found was that this kind of a new idea of what could be wrong with the old tissues. Sometimes if there's damage, you need to heal it. It might be that the old tissue, in this case in the intestine, is capable of driving the very first steps of the regenerative program extremely well. However, they get stuck in it and don't find ways out of it into normal differentiation, which after all is necessary for the function to be restored and the weight to be again restored after that. So I'm out of time, so I have to go into the thank you slides, and I'll be happy for questions.
Thank you so much, Pekka. One very quick question from Marco.
Very nice talk. Just a quick question. What do you think there is a role for mechanical stress in epithelium aging in the intestine?
Yeah, that's a great question, and we have. This might be a long answer, so I'll talk to you later. But yes,
Great. Thank you so much.
Everybody can join on it. It's a long answer, but it definitely is important.
That's great. Take it later. Thank you so much, Pekka. Really appreciate it.